Hello, everyone, and welcome to today's live broadcast, Novel Roles of Actin Cytoskeleton on Mitochondrial Dynamics and Homeostasis. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Andor. For more information on our sponsor, please visit andor.com. I would like to remind everyone that today's event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many of your questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of your presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. Today's presentation is educational and thus offers continuing education credits. At the conclusion of this webinar, please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I would now like to introduce our presenters. Dr. Raj Chakrabarty, Research Associate at Geisel School of Medicine at Dartmouth. Dr. Claudia Florindo, Product Specialist, Life Sciences and/or Technology. And also joining us, we have Meredith Price, Product and Business Manager at Amaris. For complete biographies on our speakers, please visit the Biography tab at the top of your screen. Dr. Chakrabarty, Dr. Florindo, Meredith, you may now begin your presentations.
Hello, everyone. I'm Raj, and thank you, Christine, for the wonderful introduction, and thanks to Claudia for giving me the opportunity to present my work that I've been doing in the Higgs lab. So what you can see in the screen today I'm going to talk about is the role of actin filaments, which is one of the cytoskeletal proteins in the cells, and the role it has on the mitochondrial dynamics. Now, from the image that you can see over here is actin filaments are everywhere. So whatever you see in green are actin filaments. But specifically, I'm going to talk about the actin filaments that I'm labeling over here, which is in the perinuclear region, and that dynamically comes up and then goes down again. So dynamically, actin filaments assemble and they depolymerize, and this polymerization and depolymerization of the actin filaments in this perinuclear region, what you can see here, affects mitochondrial dynamics and mitochondrial division in a unique way. And I should say that this image that you can see over here is one of the first images I took on the Andor scope when it was demoing in our lab, and this is our best image that we got, and we published this a couple of years back. So I'll start with introducing the first two main players in today's talk, actin and mitochondria. Actin, as I told you, is a cytoskeleton protein. So in cells, actin exists in monomers and in filamentous form. And it can spontaneously assemble into filaments, but this is kinetically unfavorable. So the monomers can assemble into filaments, and it has to go through this step called nucleation, where three actin monomers form a nucleus. And this is unfavorable because of the high dissociation constant of this nucleus back into monomers. So cells have devised various ways in order to stabilize this nucleation step and also help in the elongation step, thereby forming filaments. And two very important proteins that are required for this process are a group of protein called formins. So what formin does is it can stabilize this nucleation step, as you can see over here. And additionally, it can stay at this path end and thereby help adding monomers and extending these filaments. It also helps in the elongation of the filaments. A somewhat different approach is taken by another group of proteins called the Rp3 complex. So this is a complex of about 6 or 7 proteins that can get on the existing mother filament, on the sides of the existing mother filament, and then it can extend these filaments as a typical 70-degree angle. And unlike the formins, what you can see here is it needs an existing mother filament for its actin assembly. So what I'm going to talk about today is a typical role of a typical formin protein that we found out in mitochondrial dynamics. And for the time's sake, I won't be explaining the role of Rp3 complex in mitochondrial dynamics, which, by the way, has a very different role in the mitochondrial dynamics. Now, the second player is mitochondria. This is a very textbook image of a mitochondria that you will recall in your biochem classes. So mitochondria is always pictured as these kidney-shaped organelles. It has two elaborate membranes, the outer mitochondrial membrane, which is black, and the inner membrane, which is thrown into folds of cristae. And these folds of cristae harbor the electron transport chain complexes that help in the generation of ATP. But if you look at the cells, it doesn't look like these pill-shaped singular organelles. Contrary to what I showed you in that image, when you stain mitochondria in cells, it comes in different shapes and sizes. Some of the mitochondria in the cells are elongated and in network. Sometimes they are fragmented. And even in lower organisms like yeast, it has its definite shape and size. So how does mitochondria modulate its shape and size? The inherent ability of mitochondria to divide and fuse generates all these different shapes and sizes of mitochondria. So what you can see here in the screen is to your left is elongated branched mitochondria, and towards its right is small mitochondria. And this elongated mitochondria can divide and generate these smaller mitochondria. Contrary to this, the smaller mitochondria can also fuse back and generate these elongated mitochondria. And these cycles of division and fusion is very well coordinated in the cells. Now, I should tell you that mitochondria is a unique organelle which cannot be generated de novo. It has to be generated from a pre-existing mitochondria. So the cycles of division and fusion is very important for mitochondrial maintenance. These divisions and fusion of mitochondria is enacted upon by its own repertoire of proteins. One of the important proteins for mitochondrial division is Rp1. So what is Rp1? So Rp1 is a dynamine family GTPase protein that is in the cytosol, and we found out that a population of it also exists on the ER. But during mitochondrial division, it can get onto the site where the mitochondria needs to divide and then help in the mitochondrial division. One of the important proteins of fusion is called OPA1, which I won't be going into details for today's talk, but just remember that this protein is also important for the inner membrane fusion of the mitochondria. Now, why do the cell need to divide and fuse mitochondria? Is it all needed? The short answer is yes. It's needed for the normal physiology of the cell. On one hand, fusion of mitochondria helps in the complementation of the mitochondrial DNA. And this is very well required for the maintenance of mitochondrial function. What fusion also does is it dissipates the metabolic energy. Now, for its proper functioning, mitochondria has a definite membrane potential. And by fusing two mitochondria, a part of the mitochondria can regain the metabolic energy that it needs if it's damaged. So what are the utility of the fission process or the division process? Now, a very important function of mitochondrial division is in mitophagy, where a deranged mitochondria is turned over by specific autophagy known as mitophagy. And for the autophagosomes to form around these mitochondria, it needs to be broken down into smaller particles. And that's why you need the mitochondria to divide and get rid of those particles. Another important function of the mitochondrial division is distribution of the mitochondria. Now, this is especially relevant in the neurons, where in the longer parts of the axon and the dendrites, the elongated or the elaborately branched mitochondria cannot get there. So you need the mitochondrial division in the cell body of a neuron for it to be facilitated distribution along the axon and to the terminal parts of the axon. What people have also shown is this division of mitochondria is required for the cytochrome C release and apoptosis. And for us, the most important of the mitochondrial division function is in the growth and inheritance, where there is a stimulated mitochondrial division just before the cell division, where these divided mitochondria have to be distributed into two daughter cells so that each of the daughter cells have equal number of mitochondria, and this is very important. So I'll start with a very basic model of the mitochondrial division. So what would it have? As I told you, it will have this particular protein called Rp3, which is a dynamine family GTPase that's in the cytosol, and it will obviously have mitochondria. So division starts with a division signal, and by the end of the talk, I'll let you know what this division signal actually means. But the division signal, as of now, is the site where the mitochondria needs to divide, or where the Rp3 will finally assemble to divide the mitochondrial outer membrane. So once this pre-existing site is designated, Rp3 comes along and starts oligomerizing at these sites. And upon GTP hydrolysis, this squeezes the outer mitochondrial membrane and finally helps in the mitochondrial division. Now, this is a very simple way of explaining how mitochondria divides. And as my talk goes, I will add levels of complexity to this apparently very basic model of mitochondrial division. So our interest in the mitochondrial division and actin dates back to this study from the lab, where, as I told you, one of the formin proteins called INF2, which, let me remind you, is a protein that can help assemble actin filaments by stabilizing the nucleation step and also by helping in the elongation step. And when we knock down a particular formin called INF2, what we see is this elongated structure of the mitochondria, as you can see over here, compared to the control cells. And this was intriguing to us, especially because this formin is not on mitochondria. So where is this formin located? This formin has two different isoforms, and these are the domain architecture of a specific formin, and I won't be going into the details of the domains, but just to be simple enough, the FH1 and FH2 domains, which are pretty well conserved in all the formins, is used for the actin assembly purpose, and this DIT and the DAD region is used for its autoinhibition. Now, INF2 in the cells exists in two different isoforms. One of the isoforms, which we label as CAX because of the terminal cysteine, and the other isoform, which we label as non-CAX, does not have these terminal amino acids. Because of the presence of this cysteine, it gets modified and then it gets lodged onto the ER. So what you can see here is INF2 CAX, which is completely ER bound. And if you express the non-CAX in the cells, contrary to what you see over here, it's completely cytosolic. So this is where our intriguing fact is that if you deplete cells of INF2, which is either on cytosol or on ER, you get an elongated mitochondria. So we were fascinated by this, and we wanted to understand how was INF2 mediating mitochondrial dynamics. And let me just information that INF2 is mutated in two human diseases. One is a kidney disease called focal segmental glomerulosclerosis, and the other is terminal neuropathy known as Charcot-Marie-Tooth disease. So, as I told you, INF2 is localized on the ER membrane, and for today's talk, I'll be specific on the CAX isoform. And what we can do is INF2, like many other formins, is also autoinhibited, and we need to activate this protein. And a unique way that we can activate this protein is by raising the intracellular calcium. So once you increase the cytosolic calcium, either by bringing in calcium from the extracellular source or by allowing ER to release its calcium, INF2 can get activated. And so we use both these techniques. So the first one we use is by bringing in calcium from the extracellular source. So these are some of the calcium stores in the cells. The cytosol mainly has about 0.001 millimolar of calcium, whereas the ER has about 0.5 millimolar of calcium. The resting mitochondrial calcium is also very similar to the cytosolic calcium levels. And the media that we image the cells in has over 1 millimolar of calcium. So when you add this drug called ionomycin, it simply binds calcium in the extracellular medium. And because of this huge concentration gradient of calcium across this membrane, it can bring in calcium into the cytosol. And this raises the cytosolic calcium. And we can image this and find a sharp spike of cytosolic calcium, which goes down by about 100 to 150 seconds. What we also notice is this influx of calcium also induces an efflux from the ER stores. And we can measure this if we target a specific ER-bound calcium sensor. We can see that there is a sharp spike downwards of the ER calcium efflux compared to the DMSOs. So either by releasing it from the ER or by bringing it from the extracellular source, we can activate INF2. And once it's activated, the first thing it does is make this nucleus of actin and then elongates the actin filaments on the ER. And we can see this nicely by light cell imaging. So what you can see here is, so just a clarification that all the movies that I show here are taken on the Andor scope, which helped us a lot, and except one of the movies, which is a Supervis movie. So this movie in which these are U2OS cells that we have labeled for actin filaments in green over here. And this is the perinuclear region, which doesn't have a lot of stress fibers, and that's why we image in this region to get a better hold of the actin filaments that come up. The red marker is an ER marker, so it stains ER. And what you will see over here is once we add the drug, there is this burst of actin filaments which overlay with the ER marker, more or less, because INF2 CAX is mainly on the ER. So we see this burst of actin filaments that assembles and deassembles, and it's a very dynamic process. And we can measure this in this graph over here. So where we add ionomycin, we see a curve of actin filaments go up, and it goes down by about 100 to 150 seconds, which, let me remind you, is typically the similar time that we saw of the cytosolic calcium spike. So it's pretty much in that same ballpark that the actin filaments form and disappears. So this, I told you, is a specific function of INF2. So we tested this out in our INF2 knockout CRISPR cell line, in which, in the same way, we have these INF2 knockout cells labeled for actin filaments in green. And when we do a similar treatment like the previous white light cells, you don't see any visible actin filaments that comes up in this perinuclear region, which suggests that this very specific function is pretty specific to INF2. And we can quantify this over here. So the other thing that we noticed before I joined the lab is when we treat the cells with ionomycin, on the other hand, we raise intracellular calcium and activate INF2, we see this Rp3 oligomerize on mitochondria. And if you label this with the actin filament curve, what you will see is the actin filament curve precedes the Rp3 oligomerization. And what the previous postdoc also found that if you inhibited this actin filament assembly by using a small molecule inhibitor of the actin polymerization called LAT-A, so what LAT-A does is it sequesters the actin monomers and does not allow the filaments to form. So if you add the drug simultaneously with your stimulus, you don't have any actin filament assembly, and then the Rp3 fails to get onto the mitochondria. And as I told you that this actin filament assembly is dependent on INF2. And consequently, what we also notice is when you add ionomycin, you have a lot of mitochondria dividing. But if you inhibit this actin filament assembly either by using this small molecule inhibitor called LAT-A or by inhibiting this important protein called Rp3, both of these completely inhibit this stimulus-induced mitochondrial division. So from all these data, we came up with an hypothesis or a model in which we tried to explain how INF2 was doing its job on mitochondrial division. So mitochondria, from this wonderful study in the science paper, is known that mitochondria always divides at the ER interface. So we'll always have this ER that are mitochondrial divisions occurring. And upon INF2 activation, there is burst of actin filament polymerization on the ER that helps in the productive Rp1 oligomerization on the mitochondria. Because I showed you from the previous data that if you don't have this actin filament assembly, the DRP oligomerization fails on the mitochondria. And this DRP1, under its constrictive force, is dividing the mitochondrial outer membrane and thereby finally helping in the mitochondrial division. But just from this model, what I figured out that there is a gap of knowledge right over here, and there might be other things happening between actin filament assembly and DRP1 oligomerization, and this is where I started to work on this project. What we noticed is, since we are raising the intracellular calcium, we first found out that there was an influx of mitochondrial calcium upon this treatment. So this is also one of the movies that we did. This is a fast acquisition movie where we imaged every about 110 milliseconds, and we could see the difference between two very fast dynamic events. So this is an U2OS cell in which we labeled the actin filaments with green, and the mitochondrial calcium is labeled in red. And once I play the movie, what we were going to see is the actin filament assembles, and there's a time gap after which the mitochondrial calcium rise, which will be evident by the increase in fluorescence of these red mitochondria. So I play the movie, I add ionomycin at this point, the actin filaments come up, and then the mitochondrial calcium rise. So there was a definite time gap between the actin filament assembly, which happens right at this point, and then the mitochondrial calcium go up. So this was very intriguing to us. Now, we wanted to confirm this just because mitochondria is an excellent calcium buffer. So it might just be that there is a rise in the intracellular calcium in the cytosol, and then the mitochondria simply is buffering up this calcium. So, but before that, we trace out the events in its sequence, and we image these in the fast kinetics mode. And what we saw is these, after the stimulus, after adding ionomycin, the first thing we see is the rise in the cytosolic calcium levels, which is closely followed by the actin response, and then which is followed by this mitochondrial calcium rise. So we could delineate these three events kinetically. Though it's very fast and dynamic, we still could delineate it. And we figured out that the actin filament assembly follows the calcium and is dependent on this calcium rise, and then the mitochondrial calcium goes up after the actin filament assembly. Now, we tested this out in a different way. So what I told you is we add ionomycin, which brings in calcium from the extracellular source, and also in a process, it also helps in the ER calcium efflux. Now, we wanted to test this in a more physiological relevant pathway in which we add an agonist that can specifically allow calcium efflux from the ER. So nothing comes in from the extracellular source, but everything comes out of the ER. So one of these ligands is called the histamine, and you have certain groups of receptors called the histamine receptors. So once histamine binds onto these histamine receptors on the plasma membrane, it starts a chain of events and generates a small molecule called the inositol triphosphate, or IP3. This IP3 can get onto or bind onto IP3 receptors that's specifically on ER and allow ER calcium release, which raises the cytosolic calcium levels. So the difference from the ionomycin treatment is here we have specified the calcium source, which is only ER and not the extracellular medium. So we imaged cytosolic calcium, actin filaments in green, and mitochondrial calcium in red. In a similar way I showed you in my previous images. These are same cells expressing all these three markers, and we're going to image them at a speed of 110 milliseconds a frame. And what you will easily be able to see is there is a rise of calcium first in the cytosol, which is followed by the actin response, and which is followed by the mitochondrial calcium uptake. What's also intriguing to see is, so this region here, this is the nucleus of the cell. And this region here is the perinuclear region, which is studded with the ER. And you will see that the calcium rises first in this region and then spreads along in the whole cell. So I'll play the movie. I'll add histamine right over here, and then you can see it rises over here and then spreads out into the whole cell, which is followed by the actin response in green in here and the mitochondrial calcium uptake. So I'll play the movie once more. We add histamine right over here, the calcium comes up, the actin comes up after that, and then the mitochondrial calcium go up. So even when we specify the calcium source, we see a similar trend where the cytosolic calcium goes up first, followed by the actin response, and followed by the mitochondrial calcium uptake. So we questioned whether this mitochondrial calcium uptake is somewhat dependent on this actin response, or is this a passive response for the mitochondria to buffer or mop up a lot of calcium that's coming out of this ER. And we wanted to do this in a couple of ways. First, we use this drug called the trenfluramine, which, let me remind you again, it's basically an actin monomer sequestration drug, and it's very fast, so you can add it as you add your histamine or inomycin, and this drug will sequester actin monomers and will not allow the actin filaments to form. So here is the curves of the control cells, where you have the actin filaments labeled here in green and mitochondrial calcium labeled here in red. And contrary to what I showed you in my previous slides, where the actin filaments were preceding the mitochondrial calcium uptake, these seem to happen at the same time just because of the time resolution. So these images were taken every 15 seconds. So you can understand that both these events are now seemingly at the same time. But if we tease apart and image faster, you can see the difference between these two events. So we added this drug, and we could sequester the actin filament assembly in this blue curve. And what we also saw is these cells in which the actin filament assembly was hampered, the mitochondrial calcium uptake was severely hampered. So it seems that this actin filament assembly was somehow helping in the mitochondrial calcium uptake. We confirmed this in our INF2 knockout cells, which, as I told you, does not have this actin filament assembly. And in these cells, the mitochondrial calcium uptake is severely reduced. And we can rescue this completely if we overexpress the INF2 tax, which seems to get back this mitochondrial calcium uptake. So from these data, it was apparent to us that this INF2-mediated actin filament assembly was somehow promoting the mitochondrial calcium uptake. So where was this mitochondria getting the calcium from? I won't go into the details of the data, but we found out that the calcium that gets into the mitochondria was coming from the ER and not the extracellular source when we added inomycin. So the question now is, how does actin filaments facilitate this ER mitochondrial calcium exchange? Now, this is a field which has been studied for pretty long time now, and it's pretty well documented that the ER docks with mitochondria at several places, and there are bona fide proteins that can bridge these ER mitochondrial contact sites, which help in transfer of not only calcium but also lipids and other substances. So we wondered if actin filaments were promoting ER mitocontacts to facilitate this calcium transfer. So to answer this, we did some thin section EMs, and so what you see here is it was wild-type cells treated with inomycin for about a minute, which is supposedly the peak of the actin filaments, and the same treatment done in the knockout cells. And readily what you see is in the wild-type cells, the ER is labeled over here, and the mitochondria, they seem to be tightly bound to each other. But In the knockout cells, these ER and mitochondria are pretty much far apart from each other as zoomed in here. So what you can see is the ER is over here, and these are the two mitochondria with the cristae, and they form these close contacts pretty much in the wild type cells. But this distance here is pretty wide apart, and it's believed that distances of 10 to 30 nanometer is required for efficient transfer of materials from ER to mitochondria. And these distances are well over 100 nanometers. So we quantified this and found out that on ionomycin treatment in the wild type cells, there was about twofold increase in the close contacts between ER and mitochondria, whereas in the knockout cells, you don't have this increase because actin filaments were not formed. We wanted to test this in a different way. So I told you that there are pairs of protein on ER and mitochondria that help form this molecular bridge. And let me remind you, these are not very static bridges. These are very dynamic structures which can form and disentangle again. So it's required on certain parts, and when it's not required, it's disentangled from each other. So what we wanted to do is express a couple of these proteins. And if actin was facilitating this ER mitocontact to promote the calcium transfer, then by overexpressing these proteins in the knockout cells, could we get back the calcium uptake phenotype? So what we did was we expressed these two proteins called VAB-B and PTP-IP51. Now, let me remind you that if you overexpress both of these together, the whole of the mitochondrial network falls on the ER because of the over-tethering of ER and mitochondria. So what we did was express either VAB-B or PTP-IP51 singularly in cells. And it's pretty well documented that if you just express one of these partners, you have almost 2 to 3-fold increase in the ER mitocontact basally, even before in treatment. So if our hypothesis was correct and actin was facilitating this ER mitocontact, then by expressing these proteins in the INF to knockout cells, we could artificially tether the ER and mitochondria. And now if we stimulated these cells, calcium should flow in the mitochondria. So here are the images for whatever I say. The first lane is the wild type cells with the myocalcium here. And upon stimulation, you have a rise in the myocalcium levels over here. In the INF to knockout cells that have been transfected with a control marker called TEC61, which lodges itself on the ER, you don't have much of these calcium transfer happening. But if you overexpress PTP-IP, which is on mitochondria, shown here in green, or VAB-B, which is on ER, shown here in green, both of these could rescue the mitochondrial uptake phenotype in the knockout cells, suggesting that, and this is the quantification. So these results suggest that if you can rescue just the tethering function of the ER and mitochondria, the calcium flow is rescued even in the knockout cells, suggesting that actin filaments were actively playing a role in tethering the ER and mitochondria to promote these calcium transfers. So let me modify this working model a little bit. So we'll add some information over here. So this is what I showed you that the previous belief that we have that actin filaments help assemble the Rp1 that aids in the mitochondrial division. Now what I found out is there is an additional step. So these actin filaments that form on the ER also facilitate first the ER mitocontact. And this ER mitocontact helps in the calcium transfer from ER to mitochondria. Now let me remind you again that these mitochondria are excellent calcium buffers. So it's simply that your ER is releasing a lot of calcium and the mitochondria is mopping it up. So the next question is this calcium transfer at all required for mitochondrial division? To answer this question, we try to limit the calcium transfer in the mitochondria. And one way you can do that is by inhibiting this complex called the mitochondrial calcium uniporter. So calcium comes in the matrix of the mitochondria through this calcium uniporter that's on the inner membrane. And 90% of the calcium comes in through this uniporter. And once you block this importer by doing siRNA-mediated knockdown that you can see over here, what we saw is the mitochondrial calcium uptake in these MCU knockdown cells was severely reduced. But what was good for us to know that these cells all still had the actin response. So we have a scenario where the calcium efflux from the ER is there, the actin response is there, but the mitochondria is not getting calcium. So in this scenario, is the mitochondria competent enough to divide? First, we did not do any stimulation, but we just wanted to measure the fission events in the MCU knockdown cells. And we saw a very slight but significant difference between the number of fission events in the MCU knockdown cells. And interestingly, if you add ionomycin, then the number of fission events, as I showed you in one of my previous slides, goes up about threefold than the control. But in the MCU knockdown cells, these mitochondria do not divide, which suggested that this actin-mediated calcium transfer into the mitochondria is required for the mitochondrial division. So now that we know that this calcium transfer is required for mitochondrial division, the apparent question was, how does calcium influence mitochondrial division? To answer that, we did some close imaging on the Dragonfly, and this is these are all done on the Dragonfly scope. So what we noticed is if you observe mitochondria, mitochondrial calcium, and a mito-BFP marker that just labels the mitochondrial matrix, upon calcium influx, you'll see that there are some bead-on streams that form on mitochondria, or constrictions that form on mitochondria, some of which relax back and some of which result in division. Let me play the movie first. So once I play the movie, the calcium goes up, and then you can see these bead-on streams or constrictions that happen. And there are two fission events happening right over here and one over here, and some of these relax back. So our first hypothesis was that this Rp3 was coming around on the mitochondria and squeezing parts of the mitochondria, and thereby you see these bulged out regions, which are right across Rp3. So we repeated this experiment with a Rp3 knockdown cells. And contrary to what we thought, there was actually a lot of these constrictions that were forming after calcium influx, and these were not dividing, which suggested that you need Rp3 for mitochondrial division, but these constrictions were Rp3 independent, which also suggested that these are not something coming from the outer mitochondrial membrane, but might be generated from the inner mitochondrial membrane. And we often term these as frustrated constrictions, just because you have a lot of these constrictions, but they don't result in a full mitochondrial division. Interestingly, though, when we imaged our MCU knockdown cells, which do not have this calcium uptake, from these two images, what you can see, the stark difference is we don't have a lot of these constrictions happening in these mitochondria. So these constrictions were definitely independent of Rp3, but also dependent on the calcium influx. So we suggested that calcium influx was somehow influencing these constrictions, which were required for mitochondrial division. So here's the quantification where in the control cells, you have a certain number of constrictions happening after calcium influx. Rp3, you have a lot more to start with, and then also there is a huge influx of these constrictions, and most of these do not result in mitochondrial division. But in the MCU knockdown cells, you don't have a lot of constrictions to start with, and these numbers do not go up when you add this calcium stimulation. So what do these constrictions really mean for mitochondrial division? We followed these constrictions and the mitochondrial division in the Eriscan microscope, and here what we see is the mitochondrial matrix is labeled in red, and the outer mitochondrial membrane is labeled in green. And so upon calcium influx, what you see is the inner membrane dividing first, right over here. So these are blown out images. which we tried to see if there was any tether that was there, but we couldn't find any tether of the matrix. But the outer membrane is still connected over here. And then the outer membrane finally divides in this region. So what it meant was this calcium influx, which generated these constrictions, were actually dividing the inner membrane first, which stayed divided, and then the outer membrane finally would snap off under the influence of the DRP1. So the model that we have right now over here is upon this calcium transfer, what you have is this IMM constrictions, which are independent of DRP1. And these constrictions generate a intermediate step where the IMM divides and stay divided. And this DRP1, which, by the way, also is helped by actin filaments, can produce this constricting force and divide the outer membrane. So what we found out that INF2-mediated actin filaments were promoting dynamics and division of both the inner membrane and the outer membrane. The inner membrane dividing under the influence of this calcium that was coming from ER, and the outer membrane dividing under the influence of DRP1. And both of these were regulated by the actin filaments. So with this, I would end, and I would like to acknowledge a lot of people, especially Harry, who has been a very good mentor and has given me the free run to explore all these. Dakshan, who has worked, a grad student who has worked with me on a different project, which I couldn't talk about, that involves the Rp3 complex and mitochondrial dynamics. I would also like to extend my thanks for the imaging core, Zenek, and some of the past members, especially Vika, who was involved with me in this project. And lastly, I would like to thank Andor for giving me this opportunity to speak. And with that, I would pass on the baton to Meredith for her talk.
Thanks, Raj. Hi, everyone. My name is Meredith Price, and I'm the product manager for Amaris here at Andor. I'm going to tell you a little bit about Amaris for mitochondrial research. But first, I wanted to give a quick introduction to the Amaris family of software. So we have 4 main softwares that I want to tell you about today. The first is the flagship, the Amaris software. This is the one that allows you to perform that 3D and 4D visualization and analysis of your microscopy images. And so those could be wide field, confocal, light sheet, super resolution images. We're able to open up a variety of different formats. And within the software, you're able to zoom in, rotate around, create movies, but also to do that analysis where you're performing segmentation and reporting results. Then the next one is Amaris Stitcher. This is a software that you'd use if you have a very large specimen that you want to acquire images of at a high resolution. And the problem is that to acquire at such a high resolution, the whole specimen doesn't fit within the field of view of your microscope. So you acquire many different image tiles, and then after the fact, you need to align those and fuse them into one image so that you're either presenting or analyzing just one image as a whole. And so Amaris Stitcher is built for that job, and so we have that. It's nice and easy to use. We also have 2 free softwares. We have Amaris Viewer, which is a free image viewer. It's able to open up Amaris files that you've made within the regular Amaris software. It's also able to open up all the different file formats that the Amaris software is able to open as well. And so you really can use this as a free software to view any of the 3D images that you have out there. And we do open up TIFFs. If you're interested in that in Amaris Viewer, you can find that at amaris.com/viewer, a free download. And then we also have File Converter, and this is mostly useful for people that are using Stitcher or Amaris and want to be able to perform the file conversions on different computers to save themselves some time. And so again, this is a free software that's nice and light and able to do that for you. So we have those those four members of the Amaris family, and for the rest of the talk today, I'm going to talk just about the main Amaris software. And so what can Amaris do for the cell biologists that are listening to this webinar right now? What Amaris is able to do for you is that it's offering at its core the visualization of your images within 3D. So everyone listening right now knows that the organelles within your cells, the cells themselves, and the proteins, they're actually 3D objects. And so looking at them slice by slice is not giving you that full view. And so of course we're doing that 3D visualization for you, as well as being able to look at that over time so that you can see changes. But in addition to just the visualization, we are able to perform that segmentation based on the labels that you have within your image to then report out morphological measurements. And so what I mean by that are things like reporting volume or surface area, as well as different shape measurements to determine if something is elongated in one axis or another, that sort of thing. We're also able to look at the dynamics. So if you're lucky enough to be doing live imaging, that was my background, you know that there's a number of different things that you might want to do as someone looking at a live sample. You might just want to see how the position of an object changes over time. Your mitochondria might be moving around, or they might be changing shape, and you want to see how that changes over time. And that's something that Amaris can do as well. What we see many times with cell biologists is that they're looking to get a readout of co-localization within their images as well. So we do the traditional intensity-based co-localization. You may have heard of Pearson's coefficients and that sort of thing. We're able to report those measurements, but we're also seeing many people looking at object-based co-localization. And what that means is that first you would go into Amaris and you would identify your objects just like you would to create these morphological measurements. But then once you have them for your, let's say, your two different organelles or your mitochondria that's labeled in two different ways, you have two different populations of some sort. Once you have those, then you can see, are they interacting in certain ways? Are they touching one another? How many objects are touching one another? We can also help you understand the spatial distribution of your organelles or your proteins within the cell. So for that, it's not just understanding where they are within the image, but you want to understand where they are in relation to a certain object. And so that could be the nucleus, or you could pick a specific reference point. We also allow you to understand how far away they are from the edge of the cell. And so those are some built-in tools that we have within Amaris that can help you get the exact kind of results that you're looking for to help you with your specific experiments and research. So now I wanted to show you a couple of different ways that some of our customers have used Amaris in their research. We have an example here from Jennifer Lippincott-Schwartz's lab, and Sarah Cohen was nice enough to give us the rights to show this image where you're seeing different organelles and their distribution within just one cell. And so what they did was they used a tool that's in a previous version of Amaris called distance transformations to help them understand how far away objects are from one another. So how far away are, in this case, these are mitochondria, and we have other types of organelles in the different colors here. How far away or how often are each of these organelle types touching another organelle type? And as I said in the past, we had this through a tool called distance transformation, and now we have that built into Amaris with the newest version 9.5. And then over here on the right-hand side, instead of using our surface tool like Sarah did, instead this lab was able to segment out their mitochondrial label that they have. You can see the fluorescent image on the left-hand side, and they used the filament model. And so this model is able to give you measurements for the length of the mitochondria, even if they are curvy like a snake, that sort of thing. And so you can see that model here. It's less of a blobby model and more of a column or a rope, something like that, to do that analysis and characterization. And so in this example, they were looking over time and they could see how the shape of the mitochondria changed over time. And both of these are found within our Learning Center at amaris.com, and you can look up these names to find out more about exactly what they did and then find the original paper as well.
So today I hope you've learned a little bit about how Amaris could help you with your own research. If you're interested in learning more, just let us know and we'll make sure to reach out to you. You can also go onto our website to find more as well. And now I think Claudia is going to get started, and so I'm going to send it over to her. Thanks.
Thank you, Meredith, and thank you, Dr. Chakrabarty, for your presentations. And I will wrap up presenting the Dragonfly multimode system and showing why it is an essential system, an outstanding system for imaging intracellular dynamic events. So common challenges in the imaging intracellular dynamic events include avoid phototoxicity and photobleaching. And while avoid phototoxicity and photobleaching, researchers also need to acquire the images with enough temporal resolution to visualize the dynamic of the event that they are acquiring and studying, such as either being a very fast dynamic event or a very long time-lapse experiment. Also, the accurate spatial resolution is needed to capture all the information of the biological process, and sometimes live cell super resolution it is also required. The throughput of data to increase the productivity, researchers will benefit on being able to image multiple imaging fields in a single experiment to have more data to analyze and have their experiments to being statistically significant. Allow the access of multiple imaging modalities will allow the researcher to tackle different biological problems or use different imaging technologies to answer one biological question. For fluorescence illumination microscopy, we have wide field imaging, where all the light that goes through the sample is captured by the detector, turf imaging, where the sample is imaged in an angle and only a thin Z section at the surface of the sample is captured by the detector, and the confocal techniques. Point scanner confocal, where a single pinhole discards the out-of-focus light, and spinning disk, multiple pinholes will discard the out-of-focus light. And in the next slide, I will compare point scannings with spinning disk Dragonfly. So single point scanning confocal will scan the image point by point by point, and this, as you can see, it is a very long-lasting process, whereas the Dragonfly multi-point confocal will deliver the image in an instant. Furthermore, Dragonfly benefits from Andor's EMCCDs and CMOS cameras technology with up to 90% of quantum efficiency, capturing all the photons, nearly all the photons that come from your imaging, whereas the single point scanning confocals, the PMTs, photomultiplier tubes, have up to, at the most, 45% of quantum efficiency. Dragonfly multi-point scanning confocal is based on the dual microlength spinning technology. It is a high-contrast multimodal platform. It is an instant confocal because it delivers confocal image in an instant, but it is a multimodal platform because besides of being a confocal, it also delivers laser wide-field imaging, super-resolution techniques such as D-Storm and SurfStream, and turf imaging. Dragonfly comes with the Borealis illumination system, allowing a very flat, uniform illumination across the whole wavelengths from the visible to the near-infrared wavelengths. Key specifications of the Dragonfly include up to 400 frames per second in the confocal mode, a very large field of view, and a very broad excitation wavelength and highly uniformity across the excitation wavelength with a 16-bit dynamic range of capturing the image. And this is very important because you can acquire all the signals in an image, from the very dim to the very high signals, without having imaging saturation or losing any of the low signals. Therefore, it is not surprising that the Dragonfly can be used for a very broad range of applications, such as live and fixed cell imaging, membrane dynamics, cell and developmental biology, cancer research, organoid biology, neuroscience, and single molecule localization imaging. So I will now present real-life examples of overcoming live imaging challenges with the drug. In this example, researchers were able to do a time-lapse for 48 hours using the Fushy system, acquiring images with the double color simultaneously, and even 48 hours of imaging the cell cycle dynamics, cells were still dividing happily due to the Dragonfly spinning disk system. But if you want to acquire very fast events, such as calcium imaging, you can also use the Dragonfly, acquiring images, for example, at 53 frames per second. And with the Dragonfly, you can acquire dynamic events up to 400 frames per second. But sometimes researchers need super resolution, and need super resolution in live cell. The problem is that techniques that deliver super resolution most of the times are not compatible with live cell imaging. And why? Due to very complex sample preparation, due to very long times required to deliver an image, and very importantly, due to the high energy requirements to deliver a super resolved image. In 2016, Professor Ricardo Henriques have developed SURF, which is an algorithm that delivers super resolution images that is compatible with live cell imaging. Furthermore, Ricardo have collaborated with Andor. Professor Ricardo has collaborated with Andor, and we have developed SurfStream, which is an algorithm embedded in our Fusion software, Dragonfly software, and in our cameras. SurfStream will yield an increase in the resolution between 2 to 6 times more in the final data, delivering super resolution images on the fly, in the instant, while you are acquiring. The low power requirements for SurfStream make it compatible with live cell images and can deliver images that break the diffraction limit as fast as 10 frames per second. No specific sample is required. Specific sample preparation is required, and super resolved images can be delivered deep inside cells or tissues. But the Dragonfly can also deliver other imaging modalities, such as TURF, for example, for high-resolution cell surface events, as the ones that I'm showing here, using the taline and the DLC1 genes in green and red, respectively. But if you want to have access to more resolution and doing single molecule localization microscopy, you can do that with the Dragonfly using Distorm, increasing the density power of the illumination. And here you have an example of resolving neuronal spines with the Distorm. Dragonfly can also deliver 3D Distorm data using an astigmatic lens. But also, Dragonfly can be used for deep imaging. Due to custom pinhole spacing, the Dragonfly is ideal to image deep inside cells or tissues, as in this example of this cleared vessel's brain, where 2.4 mm deep were imaged, as you can see in this video. The Borealis illumination system allows very flat illumination across the whole field of view, delivering very high contrast and very flat illumination, which is ideal for stitching and quantification. As for an example, if you want to image this zebrafish and want to image it all without noticing the tiles, due to Borealis illumination, you can have the image of the whole fish, but you can acquire this fish with the 60x or the 100x lens and see the details, for example, that you are interested in study in the eye. You see the details in the eye, but you also see the whole phenotype of the fish, stitching the whole fish together without any noticing of the adjacent tiles. Fusion, the Dragonfly software, also allows real-time deconvolution, so you can increase the signal-to-noise ratio of your image and also the resolution of your image and seeing what is happening in real time with Fusion clear GPU deconvolution. The Dragonfly comes in two models, the Dragonfly 500, a complete solution delivering all the imaging modalities that I had just mentioned, and the Dragonfly 200, a more compact solution that is offered in the upright and inverted systems. So going back to the first slide, due to very gentle illumination, the Dragonfly allows you to image, avoiding phototoxicity and photobleaching, and also delivering the temporal resolution that you need to acquire the dynamic events, either very long time-lapse or very fast dynamic experiments. The Dragonfly can deliver real-time live cell super resolution, and therefore you can acquire the spatial resolution with all the information you need to capture your biological processes. Due to the stitching capability, the ability to acquire multiple tiles, multiple fields, it will allow you to increase productivity of your live cell experiments. The access of multiple imaging modalities, either confocal, wide field, storm, turf, surf stream, allow you to tackle the same biological problems in a different angle or multiple biological problems, and therefore delivering all you need for your imaging biological experiments. And this is all for now. I'm very happy to take any questions. Thank you all.
Thank you, Dr. Florendo and Dr. Chakrabarty, for your informative presentations. We will now start the live Q&A portion of our webinar. Now to our audience, if you have a question you would like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. Our first question is, Dr. Florendo, let's start with you. Can you give me a real-life example that demonstrates the productivity of the Dragonfly?
Thank you for your question. And I imagine this comes in line with the presentation of the spinning disk and the productivity of using a spinning disk versus a point scanner. So if I think about it, I remember that in April, we have posted on our Twitter account, a 3-color image that had more than 150 tiles and I think more than 80,000 images. And all of this was acquired in under 30 minutes. And so I remember more or less these numbers. I think it's more than this. I'm not completely sure. But if it's 8,000 images, more than 150 tiles, 3-color images, it is very strong productivity of the Dragonfly. Thank you so much.
And thank you. And Dr. Chakrabarty, let's come over to you. Does mitochondrial calcium uptake regulate Rp3 oligomerization on mitochondria?
That's a great question. In fact, I didn't have time to show the data, but what we did show is the MCU knockdown, which restricts the calcium entry into the mitochondria. In that scenario, the Rp3 oligomerization on the mitochondria was absolutely fine. So it suggested that it has independent pathways, and the mitochondrial calcium per se does not determine the Rp1's ability to get onto the mitochondria. But definitely the cytosolic calcium levels do influence, and there is a pathway for that. So thank you. It's a good question.
Thank you. And let's go ahead and have you answer this question as well. Does constriction and or IMM division require an active ETC?
That's a great question. In fact, we have done this. To cut the long story short, it does influence. So if you block the electron transport chain using various inhibitors like the complex 1 inhibitor or the complex 3 inhibitor, antimycin and botanon, we see a significant decrease in the number of constrictions and as well as the number of divisions. So yeah, you need a functional electron transport chain complex to be running to facilitate those constrictions that happen after calcium stimulation.
Thank you. And Dr. Florendo, let's wrap with this final question. Is it possible to use photomanipulation tools with the Dragonfly?
Yes, it is. We at Andor also supply photomanipulation tools that can be used with the Dragonfly or with other microscope systems, namely the Micropoint and Mosaic. Micropoint is high-power laser for high-power applications, such as DNA damage, and Mosaic is a device more suitable for optogenetics. It is extremely fast and allows the simultaneous illumination of multiple regions. And if you want to know more about these photomanipulation tools, go to Andor website or contact one of us. And thank you very much for your questions.
Thank you, Dr. Florendo, and also thank you, Dr. Chakrabarty, for your time today and your informative presentations. Do either of you have any final comments for our audience?
I could say that I would like to thank Dr. Chakrabarty and Meredith for giving the talk, and especially Dr. Chakrabarty to show us the importance of his mitochondrial research, and also to show us how does the Dragonfly is useful to do his research. And I would like to let you all know, if you want to know anything more about the Dragonfly of Amaris, please contact us at the Andor website. Thank you all.
And thank you, Dr. Florendo. Thank you. Now, before we go, I would like to thank our audience for joining us today and for their interesting questions. Those questions we did not have time to answer live today, and additionally, those submitted during the on-demand period, they will be addressed by our speakers via the email address you provided at the time of registration. I would like to once again thank Dr. Raj Chakrabarty, Dr. Claudia Florendo, and Meredith Price for their time today and for their important research. We'd also like to thank LabRoots and our sponsor Andor for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. You will now be redirected to the registration page for our next Andor webinar titled Surf and Turf, Simultaneous Spatiotemporal Super Resolution and Multiparametric Fluorescence Microscopy, presented by Thorsten Wolland, Professor of Biological Sciences and Chemistry, National University of Singapore, and Dr. Alan Mullen, Product Specialist for Microscopy Cameras at Andor Technology. This webinar will broadcast live July 16th at 8 a.m. Pacific Time, 11 a.m. Eastern Time, 5 p.m. Central European Summer Time. We hope to see you there.